Can
okay. Go ahead.
Yeah. Okay, can I start?
Yes, please start.
Okay, so first of all, I would like to thank organizers for giving this opportunity. I'm Chikara Furusawa from Riken and the University of Tokyo, Japan. So today, I'm going to talk about our analysis of high throughput bacteria's experimental evolution. First, I would like to present my background. One important characteristic of biological system is its ability to adapt and to evolve to various environmental changes. A question to be addressed here is how many degrees of freedom are necessary to describe such flexible adaptation and evolution, and how we can extract such macroscopic variables. As you know, biological system consists of huge number of components like a protein metabolite and so on, and thus it is an intrinsically very high dimensional system. However, when we consider complicated cellular functions like its replication, dynamic changes of amount of such huge number of components need to be changed somehow very coordinated manner, and such correlated dynamics can reduce the number of effective degree of freedoms. That is, phenotypic changes can be constrained on a relatively low dimensional dynamics. The emergence of low dimensionality in biological dynamics, such as gene regulatory networks or protein foldings, have been studied theoretically by including us. And however, for evolutionary dynamics, such constraint on low dimensional dynamics remains unclear, especially experimentally. So with this background, in this study, we performed experimental evolution of E. coli cells under various environmental conditions to quantify the phenotype genetic changes and clarify the dimensionality and constraint in phenotypic evolution. That is, starting from one parent E. coli strains, evolutionary dynamics and the various environmental conditions are traced, and phenotypic changes like transcriptional changes and genetic changes are analyzed. And by using this data of the evolutionary trajectories, we expect to understand how many degrees of freedoms are necessary to describe the possible phenotypic changes and how we can describe the dynamics of adaptive evolution by using small number of macroscopic variables. Because such experimental evolution is very tough work and labor intensive, we developed a fully automated system designed for the experimental evolution, which consists of liquid handling system placed in the clean booth and connected to the shaker incubator and microplate reader. Using this system, we can now maintain more than 10,000 independent culture series in a fully automated manner, which can provide a large amount of the data on the dynamics of adaptive evolution. What wet biologists need to do is just supply the tip of the new medium and so on, and all other things are done by this automated system. For the first round, we performed a systematic experimental evolution of E. coli cells using the 95 different stressors, including the various inhibitors like a cell wall synthesis inhibitor, protein synthesis inhibitors, replication inhibitor, and surfactant metal or oxidative stress inhibitor, and others include the various compounds that inhibit the growth of E. coli cells. This slide shows the method for experimental evolution we used. The cells were cultured in a synthetic medium with various concentrations of the stressors. And every 20 hours from a well with the highest concentration of the stressor in which cells were able to grow, cells were transferred into the fresh medium with various concentrations of the stressor. By iterating this daily propagation, we could obtain the cells that can grow higher concentration of the stressor. The changes of the stress resistance was quantified by minimum inhibitory concentration, MIC, which is around here. This figure shows one example of experimental evolution under the sulfasoxidazole, the kind of the metabolic inhibitor. Here, x-axis shows time. We performed the 26 days of the propagation, which roughly correspond to the 300th generation of E. coli. And y-axis shows the log transformed minimum inhibitory concentration to the sulfasoxidazole. To check the reproducibility of evolution dynamics, we maintained 6 independent culture series for each stressor. And here, the gradual increase of MIC means that the E. coli cells acquire the resistance to the sulfasoxidazole. And this figure shows that one example of the experimental evolution result, the high throughput experimental evolution. As you can see that for some stresses, MIC increase in the stepwise manner. And for other stresses, the MIC increase gradually. We confirmed that among the 95 stresses we used, significant increase of MIC was observed into the 28 stresses. And among them, we selected 48 environment with the different mechanism for the stresses and 4 independently evolved strains isolated from the endpoint cultures. And in total, so we selected 192 evolved strains for further detailed analysis. For these 192 evolved strains, we analyzed the phenotypic and genetic changes. We resequenced the evolved strains and transcriptional changes were quantified. Furthermore, we quantified the change of the resistance to the various environment for these 190 evolved strains. That is, we analyzed how the acquisition of the resistance to one stress changes the resistance or sensitivity to other stresses. For example, in the case of the 4 resistant strains of chloramphenicol, which is a protein synthesis inhibitor, they significantly increase the resistance to the acrylflavin. So different type of the stressor in comparison with the parent strains. So this acquisition of the resistance to other stress simultaneously is called cross-resistance. The chloramphenicol resistant strains show that the cross-resistance to the 15 stresses among the 47 stresses like this. In contrast, for some stresses, the chloramphenicol resistant strains became more sensitive compared to the parent strains, as shown in these figures, which is called collateral sensitivity. We confirmed that the chloramphenicol resistant strains show collateral sensitivity to 9 stresses. We analyzed that all possible combinations of the stress environment we used, which is more than 2,000 combinations, and we found that the cross-resistance and collateral sensitivity were widely seen in the resistant strains we inspected. More than 25% of the combination of the stresses showed a significant change of the resistance or sensitivity. This result means that the change of the resistance to one stress cannot be acquired independently. Instead, the resistance acquisition to the various environments are tightly interconnected, which constrain possible phenotypic changes to relatively low dimensional dynamics. We also analyzed the change of genetic sequence in these 192 resistant strains. This figure shows the position of the identified mutation in these resistant strains, where horizontal axis is the genome position, and vertical axis is just order of the resistant strains, such as that the first 4 is the chloramphenicol resistance, and the second 4 is the rifampicin resistance, and so on. And the dot represents the point of the SNP or short index. For some resistant strains, the number of mutation is much larger than others, but we confirmed that in more than 80% of resistant strains, the number of identified mutation is less than 5. And to evaluate the effect of these mutations, we select 62 mutations that are fixed in multiple evolved strains commonly, and each of them was introduced to the genome of the parent strain. And we identified the various mutations can significantly change the resistance level to various stress environment. There are too much data to present here, but for example, the reconstructed mutant of RSSB, which controls the expression level of RPOS involving the stress response machinery of E. coli, And the introduction of the identified mutation in RSP genes resulted the increase of the resistance to the various antibiotics. And at the same time, so the introduction of this mutation decreased the resistance that became more sensitive to various stresses like a metabolic inhibitor or mutagen. And so this similar increase or decrease of resistance levels are observed by introducing various mutations. And so if you are interested in this kind of the detailed analysis, so please check our recent paper published in the Nature Communications. Next, we analyze the relationship between the resistance and transcriptional changes, which show that they are correlated well and suggesting the low dimensionality in phenotypic changes. In this analysis, we try to predict the changes or resistance of various stresses based on the gene expression profile. Here, the objective variables Y is a resistant matrix representing the MIC for 48 stresses in 192 evolved strains. The explanatory matrix represents an expression of the thousands of genes in the same evolved strains. And for this, to predict the stress resistance, we use the Pierre's R, partial least square regression. In Pierre's, both high dimensional X and Y mean that the resistant matrix and expression matrix are projected on several components with some loading. In PCA, the loadings are determined to maximize the variance of the data projected on the component, but in PLS, the component loadings are determined to maximize the covariance of U and V, which enable us to choose a suitable axis to represent the resistance matrix by gene expression. A question here is that how many components are suitable necessary to predict the resistance. And to address this question, so we use a cross-validation. In this figure, the x-axis is the number of components, and y-axis is a prediction error of the test data, which are not used for the parameter fittings. As you can see that the prediction error became the minimum around the number of the component 9 or 10, or something like this. And this scatter plot shows that the accuracy of the prediction using the 9 component of gene expression axes. This at the x-axis is log transformed relative MIC. That is a positive value means that the acquisition of the resistance, and negative value is that the decrease of resistance by collateral sensitivity. As you can see that the change of the resistance can be predicted well based on the relatively small number, only the 9 or 10 number of components. This result suggests that the possible change of the transcriptional changes is somehow constrained on relatively low dimensional superplane. And this figure shows the score of the 192 resistant strains, the first 3 components in the gene expression space. A question here is how we can interpret these axes. And to answer it, we analyze the loadings of each axis to find that the gene functions that are significantly enriched in higher loading genes. As a result of this gene enrichment analysis, we found that gene related to stress response are highly enriched in the first component, which means that the first component represents how much stress response machinery of E. coli is activated. It is very natural because that these E. coli cells are evolved under the various stresses. And the second component are related to the aerobic respiration and the membrane transport, suggesting that the second component are related to the metabolic activity or cellular growth activity. In fact, the scores of the second component are well correlated with the growth rate of the evolved strain, which supports that this second component is the growth activity axis. In the third component, genes related to the amino acids, amino acid biosynthesis, especially that the synthesis of branched chain amino acid are highly enriched. The amino acid synthesis also involves the cell growth, and at the same time that these amino acids are known to act as a stress protectant, and this component may reflect such amino acid-based stress protection. So we can also interpret the other component as axes, such as this axis correspond to the glutamic acid biosynthesis, or this axis correspond to the anaerobic respiration, and so on. And so this result suggests that in the phenotypic changes in coli, there are a small number of major axes related to the cellular function, and evolutionary dynamics are constrained on these small number of major axes. And this result also suggests that we can connect between the high-dimensional high-dimensional phenotype space, that is the expression space in which each axis represents each gene expression, and the resistance space in which each axis correspond to each stress resistance. And we can connect these two high-dimensional phenotype space through the low-dimensional feature space. This is what our data suggested. And our next project is to analyze the trajectory of this evolutionary dynamics in this phenotype space instead of analyzing just only the endpoint clones. To analyze the trajectory, so we can quantify, for example, so we can quantify the transcriptome changes with a short time interval, but such large-scale time series transcript analysis requires a high experimental cost. Instead, using the automated system we developed, the trajectory in the resistance space can be quantified easily. So here, I would like to present some preliminary results for the analysis of the trajectory in the resistance space for the evolution dynamics. In this experiment, in addition to the stress used to the selection, so we quantify that the resistance to the other stresses simultaneously. Of course, that the selection by one stresses changes that the resistance or sensitivity to other various stresses by cross-resistance or collateral sensitivity. By this data, we can describe the trajectory of the evolutionary dynamics in the multidimensional resistance space. For a simple case, we analyze the trajectory in two-dimensional resistance space of the chloramphenicol and amikacin, both of which were protein synthesis inhibitor with different action mechanisms. And it is known that these two antibiotics exhibit collateral sensitivity to each other. And this figure shows the trajectory of 4 replica evolution when the parent cells are selected by amikacin for 16 days. With the increase of the amikacin resistance by the selection, the resistance to the chloramphenicol decreased due to the collateral sensitivity. This figure shows that the trajectory selected by the chloramphenicol, in this case, is the chloramphenicol resistance increased, but at the same time, the resistance to the amikacin decreased, MHC for amikacin decreased due to the collateral sensitivity. In this figure, the trajectory by these two drug selection emerged, by which we can see that there is some trade-off line between the chloramphenicol and amikacin resistance. And the next question is whether the evolutionary dynamics is constrained on this trade-off line, or other evolutionary direction is possible. And to address this question, we are now developing the methodology to control the evolutionary dynamics by adding the feedback regulation to selection environment. Let us assume that there is a target phenotype in the resistance space, and the current state is here. And the problem is how we can design the selection pressure to reach the target state based on the current and the past state in the resistance space. Here, we can control the concentration gradient of each stress independently. This is what we can control. And using the simple feedback control methodology, in which the Jacobian matrix represents the cross-resistance and collateral sensitivity relationship. And so we adopt a very simple feedback regulation methodology. And by this experimental evolution with feedback controls, our preliminary analysis suggests that evolutionary trajectory is controllable towards the target as shown in these figures. In this case, the target state is set here, and evolutionary set here, and evolutionary trajectory towards the target can be seen. Is updated. In the right figure, the target is set here, And in this case, the evolution is relatively difficult because that this direction is also not to this trade-off line, but still that different direction from that single drug selection can be achieved. And so now, we are trying to expand this method to control the evolutionary trajectory to more high-dimensional resistance space, such as 8 or 16-dimensional space. Then we will analyze the trajectory with various initial state and various target state. And we expected that this study will make it clear that the fit state are possible, fit state are difficult to reach by evolution. By fits, we expected that by fits that we can estimate the landscape of evolvability of E. coli phenotype. Let me summarize the presentation. So here we performed the experimental evolution of E. coli under various environmental conditions. So the automated system we developed is powerful and which enable us to analyze the evolutionary dynamics under various environmental conditions and engineering conditions in a very high throughput manner. And the analysis suggests that the phenotypic changes are constrained on relatively small number of major axes which are related to the cellular functions. And furthermore, so now we are developing that methodology to control the evolutionary trajectory to a target. And so we expected that this kind of systematic experimental evolution make it possible to better understanding that it is possible to describe that predict and control the evolutionary dynamics. Finally, so I would like to thank my collaborators. Almost all experimental evolution were done by Dr. Tomoya Maeda, and all data analysis were done by Junichiro Iwasawa, and that feedback regulation and the controlling trajectory were done by Dr. Shibai. And so in this conference, Junichiro Iwasawa will present a poster. So if you are interested in the detailed analysis or detailed experiments, so please check his poster. Thank you for your attention.
Okay, thank you. I do the clapping for everybody. And please type your questions or say your questions. I don't know if there's anybody who wants to start off with saying a question. Anybody? I see a question from Michael Manhart. He says, I must have missed it. In what environment do you measure transcriptomes for each strain?
Okay, so in this case, that for the transcriptome analysis to unify that condition, so we take the transcriptome data without adding any stressors, just as a no-stress condition. Is that okay?
Does that answer your question? Okay, Anton, do you have a question?
Yes, thank you. Thank you, first of all, very much for this talk and the work. The work is fantastic from my point of view, just as we need this predictability of phenotype. But I have a question about the very last thing that you showed. When you try to evolve off the low dimensional, or I don't know how you interpret it. So when you try to evolve it towards some targets, and towards some targets, evolution was easy. Towards other targets, it was difficult.
Yes. Yeah.
So interpretation would be that, as far as I understand, that in the first case, it is possible actually to go in this low dimensional manifold or whatever of feature space. Using other coordinates, right? So you somehow go there. In the other case, it's very difficult, maybe not enough.
Yeah,
I don't know what, but it still evolves towards that. So my question is the following, although probably there's no data to answer it. In principle, the feature space itself, this manifold of the features, may change along the trajectory of evolution itself. So how much would be what you see here, evolution towards a different target? How much comes from going around in the manifold and changing the manifold itself?
Yes, actually, your point is very interesting for me. And so this is what I want to understand, actually, by using this kind of methodology. So far, so we don't know that in this case is that the manifold changes or not. But now, so we are trying to analyze this dynamics of that manifold or so evolvability landscape or something like this. By, for example, starting the many initial state and the setting the many target state. And so which direction is difficult and which direction is easy, and how it depends on the initial condition or how it depends on the history of the evolution or something like this, by which we expected that whether this kind of the manifold is stable or robust, or it also dynamically changes. This is our future work.
Okay, we have another question in the chat. Amazing talk, thank you. To what extent do you think that the dimensions describing antibiotic resistance that you found will extend to other environmental factors like nutrients, abiotic factors?
Yes, it is also interesting question. So actually, so in this case is that we mainly focus on the antibiotics, but this stress we use also includes various different factors that just, for example, that just organic acid or oxidative stress inhibitor, and the others include the many different types of the stresses. Some of them that the action mechanisms are completely unknown. So also now we are planning to expand this factor, for example, that the many nutrients and how that the nutrient condition changes and the antibiotic resistance are related to something like this. So this is our future work. So now we already have that high throughput system, so we will expand this repertoire of the stress. Thank you.
Okay, anybody else, questions? I have a question. So could you extend this to look at longer time scales?
Yeah, of course that we can so expand this for more longer time scale, but actually that's and so experimental evolution, for experimental evolution, this kind of evolution, so it is difficult to decide that when we should stop. So actually that in this case that evolution is still ongoing. But actually, to be honest, so this automated system is rather robust, but sometimes they have trouble. So experiment for the long time scale is experimentally risky. So we avoid this kind of data. We focus on the rather the shorter adaptive evolution time scale. This is my answer, to be honest.
Okay, well, I suggest we take further questions to the gather room for later. So let's thank Chikara again. And Francois, can you connect?